everyone. We're so excited to have you joining us today for the hands-on DNA activity from our June 2021 virtual Discovering the Scientists Within event. My name is Ellie and my major is biochemistry and molecular biology, and I will be passing it off to Maya, Carly, and Nicole for this activity.
Yeah, thank you for having us today, and welcome to the workshop on DNA. Please make sure you have your DNA kits handy, as well as the 3 printouts that we provided you. You should have 3 charts that were in your kit. And don't open your bags quite yet. We'll let you know when it's time to do so. But the first chart you should have will look something like this. It's a color code, and we'll explain these when we get to them. The second one might look a little slightly different than this, but it should have something that says genotype and phenotype and different traits such as eyes, hair, dominant hands. So just make sure you have that handy. And then the third chart just has a bunch of names and it's little boxes. So just have those nearby. And yeah, without further ado, we'll get started. So my name is Maya.
My name is Carly.
And I'm Nicole.
Yeah, and so we're all members of the Biochemistry Club at Oregon State University. And in case you're wondering what biochemistry is, it's a branch of science that studies the chemical processes that occur in living things. And because we're all majoring in biochemistry at OSU, we're learning a lot about chemistry and biology in school. So we want to share a little bit about what we're learning about by talking about a very important topic in biology, which is DNA. So DNA is considered to be the basis of all life on Earth. And by the end of today, we hope you understand at least one new thing about DNA.
All right, so DNA stands for deoxyribonucleic acid, and this is the basis for all life. So what do you guys already know about DNA? Feel free to pause the video, take a little minute to think about what you know, and then coming back, well, DNA contains all of your genetic information.
All right, so next we're going to have a brief history of DNA. So Rosalind Franklin was a female scientist who took some of the first images showing the structure of DNA, and she's pictured here next to a picture of one of the images that she took. And she used a fancy technique called X-ray crystallography to generate her DNA pictures. And then 2 other scientists, Doctors Watson and Crick on the right side, used her images to further prove their proposed structure of DNA, which we will talk about later. And lots of people were involved in the discovery of DNA, but these are among the most prominent.
So as you can see in this picture, this is one cell in your body. And so every one of the trillions of cells in your body needs its own copy of the DNA, which basically tells the cell what to do. So the DNA is kept in a specific compartment in the cell, and this is called the nucleus. It's the red, basically ball in the center of your cell. And so every one of your cells has its own copy of DNA, which is about 2 meters long, about the height of a typical door. And if you took all the DNA in your trillions of cells and put it all together, you would have 80 trillion meters of DNA, which is long enough to get you to the sun and back over 200 times. So we obviously have a lot of DNA because your body is complicated and takes a lot of instructions to tell your body how to make sure that you become you.
For all of that DNA to fit inside of your cells, it needs to be packed in really tightly. So the easiest way for it to do that is that it twists around a couple of times. So part of this twisting gives the DNA its signature look, which is the double helix. The double helix is made up from 2 strands of DNA that are connected and then spun around many times, forming the helix shape shown. We can zoom in on the DNA to look at its structure a little more closely. If you remember the double helix that you all just saw, you can untwist the double helix to make it seem like the backbone of the DNA, and it looks like a ladder. So now we will explore a little bit more about how DNA activates this ladder structure.
Okay, so the components that make up DNA are called nucleotides, and they each have 3 parts to them. The first is phosphates, which are on the left here, and they're molecules that like to form bonds to ribose. Ribose is a really simple sugar, sort of like glucose, which is one you might have heard of before, and it likes to form bonds to multiple phosphates. So you can organize them together in an alternating order, so sugar, phosphate, to make up a really strong sugar-phosphate backbone for DNA. And this is sort of like the sides of the ladder that we were talking about in the last analogy. And then there are what are called bases, And there are 4 different ones that connect together to make up the rungs of the ladder. And now we're going to go into a little bit more detail on the bases themselves. So there are 4 of them: cytosine, thymine, guanine, and adenine. We usually just call them by their first letters C, T, G, and A. And cytosine and thymine have 1 ring structures, and they're called the pyrimidines, and guanine and adenine have 2 ring structures, and they're called the purines. And we'll talk about why these ring structures and the separation of the 1 ring versus 2 ring is important here in a bit.
So this is the color code that I tried to show you at the beginning. So you should have this handy. You'll need it to build your strand of DNA. And if you all want to open your bags at this time, you can take a minute to do so. If you need to pause the video, that's fine, but I'll start going and showing you what you should have in your kit. So to make the backbone that we just talked about, you should have pink and white beads. And so your beads should look something like this, pink and white. And then in that same bag, you'll have these little clear connectors, which you'll need last. So you can put those to the side. You don't need those for a while. And then you should have the 4 base pairs that Nicole just talked about. So the A, C, T, and G, which are the colored beads. You have the green, blue, yellow, and red beads. And those are the base pairs. So you'll have 4 different colors of those. So we're going to build our first strand of DNA. And so our strand is going to be the letters A, T, 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 C, G, C, G. And so to build that, first we need to make the backbone so that the base pairs have something to attach to. So if you want to pull out all your pink and white beads, we're going to make the backbone. Let's see. If you want to stop sharing, they might be able to see a little better if the screen is big. So yeah, so you're going to want to build the backbone. And to do this, we have 6, or we're going to want to build it long enough to fit all those letters, the AAT, TTG, etc. So you're going to want to have 9 white beads and 9 pink beads to build your strand long enough. So basically just start attaching them. They should pop pretty easily. So just do alternating. We're making a sugar phosphate backbone. And if you're still confused, you can also refer to the chart. So on your chart, you should have, it shows that the pink is the phosphate and that the white is the sugar. So we're just making our backbone. And some of the beads don't fit super great, but you have plenty of extra beads. So if they're not fitting, you can just discard those ones and try a new bead. Some of them just are a little hard to fit together, but yeah, just work on making your alternating strand, pink, white, sugar, phosphate, sugar, phosphate. Again, you should have 9 white beads and 9 pink beads when you're finished with your backbone. And you can also pause the video if you need a little more time to do this. But I feel like I'm pretty slow, so you might be done before I'm done. That should look something like this. Okay. It's a little hard to see with the light, but yeah, pink, white, pink, white. So this is your backbone. And now we're going to build the sequence that we just said. If you don't remember it, it's fine. We'll walk through it slowly together. So our first triplet, or our first basically gene, will be AAT. So if you look at your color code, you can see where A corresponds to. A or adenosine, but again, we just refer to the first letter. So A, or adenine, is going to be blue. So take your blue bead and basically just pop it in. You're going, oh, a little finicky. You're going to be attaching only to the white beads. The pink beads will just be alternating. So your first one will be A, it's going to be blue. Again, our next letter is going to be A, so another blue one. All right, so the next one is going to be T. We have AAT, so maybe take a minute, think what color will be next in our sequence. All right, so next it's going to be green. And then our next 3 letters, or our next 3 parts of our genetic code, is TTC. So T, if you look at your chart, again, that corresponds to green. So our next one will be a green bead. So right, I should have blue, blue, green, green, and then it's another T. So at this point, you'll have 3 greens in a row. All right, so our next one is going to be C. So maybe take a second, look at the chart. What color will C be? It has to be red or yellow because we haven't used those colors yet. All right, so you're right if you thought it was red. So put a red bead on there next on your code. Again, you're only connecting to the white beads. The pink beads don't really have a place for attachment, so that's helpful. All right, so right now you should have all the colors, use them at least once. So you have your CG, and then our last two ones are another CG, so another red, yellow. And if it's a little confusing, don't worry about it. You can also pause and maybe take a minute to look at your chart a little more if it's still a little new. So yeah, right now we'll have this. And if you think about this, how is this different from the helix that we just showed you? So we take a second to remember we just had that double helix. And so if you think about it, this is basically 1/2 of your strand of DNA. So we're going to have to build another strand like this so that they can attach and wind around each other to form that double helix that we just showed you in order to fit in your cell. So it needs the other half so that it can wind around itself to fit in really tightly. Yeah.
Like we just said, there are 4 bases. They are pretty picky, so when they pair, they only pair in 2 specific combinations. A goes with T and G goes with C. This is because they have to fit together in a very specific way. Notice how each pair has one 2-ring structure bounded to a 1-ring structure. So looking at the structure of DNA, we see that all the way down, the 1 rings are paired up with the 2 rings. This needs to happen because if we had two 1 rings with two 2 rings together, the structure would be uneven. It would be a lot harder for the DNA to be able to repair or replicate itself if it needed to. So the base pairing is very important to keep the DNA uniform and easy to manage.
Yeah, so now that we talked a little about base pairing, we're going to build something called the complementary strand, which we touched on earlier, so that we actually have a complete strand of DNA, because right now we still just have that 1/2. So again, we're going to want to start with building the backbone. So let's start with our alternating sugar, phosphate, and then we'll go into how the base pairing. But something that you might want to write down on your sheet of paper, such as maybe your color code, is that A and T will always pair together, and G and C will always pair together. That's if you know that, you'll basically be able to build any strand that you want. But first we'll build the backbone, and then we'll talk about which one will pair with which one. Again, you should have 9 white beads and 9 pink beads when you're finished. You have enough beads to build more, so after today, if you want to keep building longer strands of DNA, you're welcome to do so. Even if you were to use all these beads, clearly we have a lot more DNA in our body, so this is just a small representation. And DNA is very, very tiny, so this is also a little bit larger. Yeah, getting close. Okay, so you should have 2 strands right now, and this is going to be what we build the complement on. So let's start with this side. So we'll start with the AAT. So again, we're going to work in kind of pairs of 3, because that's how we usually you think about genes. So if you remember, A always pairs with T. So if we have AAT, the complement is going to be TTA. So it's kind of helpful if you think of it color-wise as well. So the cool colors will always correspond to each other, so the blue and the green, and the warm colors, the red and the yellow, will always be base pairing. So in your complement, you can go green, green, blue. Going to be the complement for the first 3 letters. The next one on our original strand is TTC. So the complement, maybe take a second, think about what you think it will be. All right, so if you thought it would be blue, blue, yellow, that is correct. So we're going to have it A corresponds with T, another T, so we have another A. And then our original strand, our C, will always correspond with G, which is yellow. All right. Now we have our last 3 letters, which in our original strand is GCG, or yellow, red, yellow. And the complement will be GC or CGC, so red, yellow, red. Just do your best. These beads are a little tricky. They pop out. Okay, so not quite done with the strand because now we have to actually connect it down the middle, which is what these little clear pieces are for. These should be in the bag that was the yellow, or it should be the backbone beads that was in the bag with the yellow, the white and the pink beads. So they're kind of in the bottom. If you don't see them, they're pretty tiny. But we're basically just going to connect down the middle. So again, this kind of part is a little hard with the beads, but if you want to just try your best, these connectors should just go between the colors to help form the rungs. And if you need to use the table, sometimes if you press it against the table, that helps. The little clear connectors just go down the middle. And if you need more time, feel free to pause, or if I'm going slower than you, which is probably the case, sorry, you'll have to wait a minute. But yeah, just work your way down, connecting them.
You might be covering this later in your presentation, but what happens if you don't do like proper base pairing in DNA?
That's a great question. We will touch on it a little more later, but usually your body is really good at catching mistakes. So if you accidentally put the wrong base color, usually the molecules that regulate DNA replication will usually catch it and just kind of fix it as it goes. But sometimes these mistakes do go unnoticed, and that can lead to something called a mutation. But we'll leave it at that for now because we will briefly talk about mutations at the end. So great question. Yes, your body, since it makes so many copies of DNA in one day, it definitely does make mistakes sometimes. Yeah. So if your strand looks a little something like this, again, these clear connectors can sometimes pop out, so just be careful. But yeah, so this is your strand. So you're gonna have to twist it to form the actual double helix. So if you have it like this is the ladder, and then if you twist it, that's the double helix we saw earlier. Yeah. Okay. So now that we know how to build a strand of our genetic code, let's think about how one's DNA makes us uniquely who we are. We all know that we look at least a little bit differently from the people around us, even though we share 99.9% of our genetic code with every human on Earth. So to do this, we're going to consider something called genotype and phenotype. So genotype refers to all the genetic information that is inside a person's cells, while phenotype refers to the physical trait that is seen as a result of one's genetic code, such as brown hair is the result of the genetic sequence GCC.
So, like Maya said, genes lead to a phenotype in people. And genes are organized in triplets of bases, like they are on the phenotype key, and like we've been doing it on the slides. So usually there's more than one triplet that goes into something as complex as hair color, for example. If you think about it, you've seen people with blonde hair, darker blonde hair, light brown, medium brown, dark brown, and black. And there's plenty of colors in between all of those distinct colors as well. So it's a little more complicated than just one triplet, but we're going to make it simple so that you don't have to make a really long strand of DNA for yourself. So here is our phenotype key that you all should have a copy of. And yeah, we are just going to do these 4 categories in our examples.
So our example is going to be actually helping us find out the mystery of the missing sandwich. So there's been a crime, a sandwich was stolen. Luckily, a piece of DNA was found in the saliva at the scene with the sequence AGG, GCT, GGG, and TAA. And so using that nifty key that we just saw, the phenotype, the genotype, take some time to look at the phenotypes there and write down all the hair color and the eye color that you might think it's out to be. Coming back, what these correlate to would be blonde hair, blue eyes, medium height, and right-handed dominance. And so I guess somebody with blonde hair and blue eyes stole our sandwich, unfortunately. And the authorities have also saw that there was an accomplice who helped them steal the sandwich. So the witness was described as having hazel eyes, brown hair, medium height, and is right-handed. So kind of doing the reverse of what we just did, we can use the phenotypes that we are given to try and look for the genetic code that would be correlating to it. And so you can take some time to like look for each of these on your key, and then coming back, if you got TGC, GCC, GGG, and TAA, then you would be right.
Nicole, did you steal the sandwich?
My friend forgot her lunch, and so she said she'd give me a bag of chips if I didn't tell anybody about the sandwich. Sorry. So now that we know a good way to use DNA and where to find it, let's think about where your DNA comes from. So usually it comes from your birth parents, and then they get it from their parents. So essentially you have some of your grandparents' DNA. This is why some people have traits that are more like their grandparents than like their parents. And since siblings get their DNA from the same parents, they tend to look similar. But you might notice that some people look a little more like their dad while their sibling looks more like their mom. And that can be part of something called dominance, which is how strong a gene is.
Yeah, so we briefly touched on this before, but if anyone made a mistake when you're making your genetic code, that's totally understandable. I know I made a mistake or two when I was building mine. But yeah, so these mistakes are often called a mutation. And a mutation basically is a change in one's genotype. So maybe switching out one of the color base pairs for a different or the wrong color, or base pairing with the rings that do not fit correctly. And so mutations can often be harmful to an animal, maybe such as like harming this cat's ability to blend into its natural environment, which could harm its ability to hunt food. Mutations can be beneficial, which is more rare because usually random changes results in either no effect or a negative effect. If you think about if you were to write an essay and randomly change out one of the words in your essay for another word, it might make the essay sound better, but more likely than not, it's going to make the essay sound worse. And so these mutations often don't have a large effect on an animal, but they can, maybe such as making a turtle with two heads. But as Nicole mentioned earlier, usually you have a lot of genetic code that goes into making one trait. So one small change in a base pair might just go unnoticed and have no effect on the animal. But it's important to know that mistakes do happen because your DNA copies itself over and over throughout the day, and it makes a lot of copies of itself. So thankfully for us, our body is very good at catching and fixing most of these mistakes.
All right, so for a brief summary of everything that we've talked about today, DNA has the structure called a double helix. It's made up of 4 bases, A, T, C, and G, that pair in a really specific way, which is A goes with T and G goes with C. And then DNA is something that you inherit from your parents, that they inherit from their parents and their parents. And finally, a person's genotype or genetic code determines their phenotype or their physical traits.
So after today, or after you're done watching this video, if you want to take some time and build your own genetic code, you can take the time to do so. So if you pull out your chart, kind of how you would code your own DNA is basically first determine your own phenotype, decide what eye color you think you have. And if you need help, you can ask people around you, maybe decide your hair color, your height color, what type of nose shape you have. And then you can use your phenotype to genotype trait or chart. And so you look at your phenotype, then maybe take a pencil and write down what genotype that refers to. So for me, I would say brown eyes. So I would look at and write TGG down because that will make it a lot easier when you actually go to the beads. And then once you write down all your genetic code, then I would say pull out your beads and you can just use your chart and kind of just go from there. So if you want to build all your 5 traits, you're welcome to do so, or you can even build a family member, a sibling maybe, or a friend. And if you first build your own and then you build a sibling, you might see that there's only one change in the phenotype because you might look very similar to a sibling. And that's just kind of showing how we do share our DNA with our family members. Yeah, and just a quick thank you to all the other biochemistry members who helped us make this presentation. They couldn't all make it today. So yeah, so I want to send out a thank you. And then if anyone has any questions, you can always email us, or yeah, just I hope you enjoyed this presentation and that you learned at least something new about DNA.
Great. Thank you so much to the Biochemistry Club, especially Maya, Nicole, and Carly, who came here to do this activity with us. Thank you so much for coming to this event and participating with Hands-on DNA, and we hope to see you for another session soon. Bye.